In order to thrive in a given environment, an organism must be able to form memories that it can use for reference in the future. Memories of positive stimuli guide organisms towards potential gains and rewards. A raccoon finds a trash can full of food and now knows that these metal bins are potential treasure chests. Additionally, negative stimuli help organisms to be wary of potential dangers that they can encounter in their environment. Our raccoon from earlier finds that one particular trash can has a dog on the property that will give chase if he gets too close. The raccoon now knows that this trash can is potentially dangerous, but how does our furry friend know that this trash can is dangerous and not form a bad association with all trash cans? One of the most important tools an organism has is the ability to create novel memories from exposure to an unknown stimulus. But in order to do this, the brain has to be able to distinguish between a novel experience versus one that has been encountered before. A recent breakthrough in the field of neurology found strong evidence that the locus coeruleus exhibits influence on the hippocampal subfield CA3 in determining the novelty of a stimulus. Before we can start talking about the influence of the locus coeruleus on the CA3, what exactly are these things and why are they important? To start, it is crucial to understand that the hippocampus is essential for the formation of new memories. The hippocampus is a unique structure that is wired to form a loop back in on itself. One key part of this loop is the CA3 region that we talked about before. The purpose of this wiring is that when neurons within this loop fire together, their connection allows them to remember who participated in the formation of that memory. This makes it so that when the stimulus is encountered again, all of the previous neurons fire off together for the recollection of the newly formed memory. The pyramidal cells within the CA3 are required to pull this off though. If the NMDA receptors that strengthen the connection between these CA3 neurons are removed, then the organism is not able to form a quick acquisition of a novel memory. However, this does not affect recall of memories that have already been formed, showing us that this mechanism is crucial to memory formation, but not recollection. This is great for understanding how the brain codes for new memories, but does not give us a clear idea of how memories are formed more readily in the presence of a novel stimulus. To begin answering this, we have to look at what happens in the brain of an organism when it is exposed to a novel stimulus. When the hippocampus detects input from a stimulus that is not present in the organism's long-term memory, it activates a functional loop with the ventral tegmental area of the midbrain. This part of the midbrain is involved in dopamine signaling for many neurological functions. When it receives the signal that there is exposure to a novel stimulus, it completes the loop with the hippocampus by releasing dopamine. Dopamine is a neurotransmitter that helps to enhance long-term potentiation, which is the strengthening of synapse connections between neurons. Due to this, dopamine release from the ventral tegmental area, the neurons within the hippocampus are able to more readily form connections with each other for the development of a new memory. However, this doesn't help us to understand what neuromodulatory input helps to distinguish a novel signal from a known one. It was also discovered, however, that the ventral tegmental area is not the only region that is active during exposure to a novel stimulus. TH plus neurons, which release precursor molecules for dopamine, were found to be heavily influenced in the locus coeruleus by novel stimulus. These neurons were then engineered to be activated by light stimulus, a technique known as optogenetic stimulation. When these TH plus neurons were activated within the locus coeruleus, the impact mimicked exposure to a novel stimulus as opposed to stimulation of TH plus neurons in the ventral tegmental area. This shows us that the locus coeruleus mediates hippocampal memory enhancement for novel experiences, although it does not show us the impact it has within the hippocampal network itself. This is the question that we were initially talking about with our recent breakthrough. It was found that the input from the locus coeruleus onto the CA3 is essential for context-based learning. Additionally, stimulus from the locus coeruleus was not necessary in the hippocampal subregion CA1 or the dentate gyrus. Inhibiting locus coeruleus signals when a subject was exposed to a novel stimulus reduced reactivation of the cell assemblies in CA3 neurons and CA1 when exposed to the stimulus later. This inhibition was shown to cause the CA3 cell assemblies to have a more random placement than normal. The reason this discovery is so groundbreaking is that we now understand the mechanism behind how relevant stimuli are preferentially encoded into memory over incidental stimuli. This leads to potential avenues of studies in the future of memory formation. Although all of these studies were performed on rats, these results can be used to infer mechanisms behind human learning as well. How does altering the locus ceruleus impact memory formation? Could manipulation of this neuromodulatory input have an impact on the strength of the memories formed? Is it possible that this mechanism explains diseases that cause deficiencies in the formation of memories? If so, we might be able to harness this knowledge to find a solution for their treatment.